Hey guys, today we're going to be having a look at PowerPoint 5, looking at reproductive technologies. We're going to be looking more on an angle from where technology is headed in terms of genetics and reproductive technologies. And this is more with respect to your research investigations. So let's start and we'll go through what you need to know. So we're focusing on reproductive technologies. There's no particular section out of the Pearson book that this is discussed in. So we're just going to be looking at some different resources today. So by the end of the lesson, you need to be able to discuss implications of genetic screening technologies, such as preimplantation genetic diagnosis and CRISPR on reproductive technologies. Now, a note here, the syllabus states that science as a human endeavour subject matter, such as this topic, will not be assessed on the external exam, but could be used in the development of claims for the research investigation. So what that means is the syllabus give us guidelines to say you should be talking about these things. However, because it's more of the ethics and the human applications, it's not explicitly taught on the, covered in the external exam. So you may want to use this stuff, however, for your research investigation, and I'm going to just talk over those claims again. And in the back of your mind while we go through this, just have a think about if any of these stand out to you as a topic that you might want to look at for your research investigation. So these are the claims that are on your research investigation task sheet. So the ones that I've highlighted are really the ones that these would be a bit more applicable to. And you can, because the claims are quite broad, some of the stuff that we'll talk about today, you can apply to those dot points. So things like genetic screening technologies have implications on reproductive technologies. That's a big one. Bioinformatics can be used to analyze relationships. Broad, but that can be brought back to reproductive technologies. Genome sequencing and genetically modified organisms. So things like CRISPR changing the genetics of something. We will go over the research investigation in future lessons, but this is just to get you thinking about it and just kind of in the back of your mind have areas that are interesting to you. All right, so in terms of reproductive technologies, often they use the abbreviation ART, A-R-T, and that stands for assisted reproductive technology. So these technologies are treatments that are used to assist people in achieving pregnancy. There's three sort of overarching purposes to this. The first is overcoming infertility issues. So this is looking at people who aren't able to conceive and achieve pregnancy naturally, so they need to overcome fertility issues. That could be to do with the male, could be to do with the female, or it could be that there is a same-sex couple or a single person that's trying to achieve a pregnancy. The second big one is the detection of birth defects. So this would be more to do with the screening and the looking into the health of the developing baby. The last one is to do with research. So this would include things like stem cells for stem cell research and CRISPR, which are not so much for the purpose of reproduction, but using reproductive technologies to come up with advancements in medicine. So in terms of overcoming infertility, like I said, there are many couples who struggle with infertility, and that can be from things to do with the defects in the sperm. It could be that there's something wrong with the female reproductive system, that ovulation mightn't be occurring. As I said before, same-sex couples might need to utilize these technologies to reproduce. The main reproductive technologies involved in this are donor gametes, ovulation induction, artificial fertilization such as IVF, in vitro fertilization, and ICSI, which is Intracytoplasmic sperm injection. We're going to go over some of these just to give you an overview of what these reproductive technologies entail. Now, when we look at donor gametes, this is talking about a person donating their egg cells or sperm cells for other people to use. Now, donor sperm is useful in a situation where a male in the scenario doesn't produce normal sperm, doesn't produce sperm at all, has a low sperm count, or perhaps there is a high risk of that male passing on a genetic disease. so you wouldn't want to use his sperm that could potentially pass on a serious disease. It could also be used for a single woman who's wanting to get a sperm donor, or women in same-sex relationships. Conversely, the donor egg is an option where the eggs are harvested from a female, and that's used when the female herself doesn't produce eggs, she has low quality eggs, or perhaps she is carrying a genetic disease that she doesn't want to pass on. And she would still be able to conceive a child with her partner using his sperm, but using a donor egg. Lastly, sometimes there are donor embryos. So when people are going out to try and achieve a pregnancy by using things like IVF, they will often mix the sperm and egg, produce embryos, and often they end up with more embryos than what they need if they achieve a pregnancy straight away. So sometimes people choose to donate those frozen embryos, the leftover ones, to other families. So that would be a scenario where that embryo could be implanted into a female. She would be able to carry that embryo into a baby and still give birth to that child, even though the DNA has come from different parents, just to have a bit more connectedness with this baby. Okay, now ovulation induction. This is a treatment that's more around issues with a woman being able to ovulate normally. So ovulation is the process that happens each month where an egg is released from the ovary. And when the egg is released, it goes down into the fallopian tube here, and it stays here for a couple of days where if there are sperm present, fertilization can occur. For some women, if hormonal levels are out of balance or for different reasons, they may not ovulate regularly. And this is a problem because if they're trying to achieve a pregnancy and there's no egg available, then it doesn't matter how many times they might try, there's no egg present, so no pregnancy is going to occur. So with this technology, this involves hormone treatments to kind of mimic the natural process or put it into overdrive if they're trying to actually collect some of these eggs. So they give the woman hormones, it stimulates the production of follicles within the ovary, and that is going to later release an egg. If that egg is released, then that means that if sexual intercourse occurs and there's sperm cells present, this couple can now achieve pregnancy potentially. This is also used for IVF, which I'll talk about a little bit later, where the eggs are released and then collected out of the female body to be fertilized externally. So this brings me to IVF, in vitro fertilization. In vitro refers to being out of the body. So it generally occurs on a petri dish, like what you'd see in a lab. And like we just talked about with the ovulation induction, the female is given drugs, hormonal drugs, to stimulate a number of her eggs being produced. So rather than just releasing one egg, they kind of take a lot of these hormonal treatments to get a number of eggs to be released. The eggs are then retrieved. So there's different processes in place where they can go up there and collect the eggs, and those eggs are placed in a storage, and when it's time for fertilization to take place, they're added to the petri dish. Sperm are then added to those eggs, and naturally fertilization occurs like it would in the body. So as you can see in the image, the sperm cells are placed onto the egg sample and are just like in the body, the sperm cell that gets to the egg first is going to be the one that fertilizes it. Sometimes, like I said, multiple eggs might be fertilized, and that way they can end up having more than one successful zygote, which can then be implanted into the uterus. Now, this is good for couples who might have issues with falling pregnant the natural way, where the mother's egg can still be used, the father's sperm can still be used. So this embryo is completely genetically linked to the parents. but the embryo then gets implanted back into the natural mother to develop as a normal pregnancy would. The other scenario is if there is an issue with the mother being able to carry her child, this embryo can be placed into another woman called a surrogate who can carry that baby to full term. She doesn't have any genetic links to that baby, but this woman would have volunteered to be the carrier of the baby and just deliver the baby to birth and then give the baby back to the natural parents. And that's the end of her role in the scenario. Now, this technology is much the same as IVF, but the only difference is instead of the sperm sample just being added to the petri dish, the sperm cells are actually injected into the egg. It's a very similar sort of thing. The main difference is in this scenario, the sperm might have trouble breaking through the egg and actually getting into the egg. So this could be defective sperm that don't swim as good as others and have issues with getting into the egg. So it's very similar. There's still a combining of the gametes externally, but the sperm are injected into the egg. Okay, now the second sort of overarching goal of these technologies is detecting birth defects. And this one has some serious ethical implications when we start thinking about it. Now we're going to have a look at pre-implantation screening. This is a technique that we use alongside other reproductive technologies like our IVF to analyze the risk of passing on genetic conditions. Now, if we have a look at the image here, this could potentially be the embryo that we've created through IVF, and this is before it's about to be implanted into the female. Now, pre-implantation screening involves one singular cell from that embryo being removed and then being genetically analyzed to look for potential issues in the genetics of this embryo. This can be used to look for single gene defects. So this is to identify embryos that are healthy and not affected by any genes that might lead to disease. So that from sort of a gene by gene basis, or it can also be used to look at chromosomal defects. So this is to make sure that there's the correct number of chromosomes to prevent conditions like Down syndrome. So once they do that, they can analyze the genes, they can look at a number of different embryos and basically select for the embryo that has no issues and then transfer that particular embryo into the woman's uterus. So this is just a way of preventing passing on any genetic conditions or undesirable traits that may lead to susceptibility to disease prior to achieving pregnancy. Okay, now research is a really interesting one because we're kind of going from trying to achieve pregnancy to medical research and medical advancement, which is where a lot of the ethical concerns come into it, because where do we draw the line with what's okay and what's not? Now, stem cells is a really good example of this, and you might remember from year 11 that the embryonic stem cells are the ones that have the most potential to differentiate into specialized cells. That involves actually removing cells from a developing embryo, which means that they can't continue to develop, and researching their potential to differentiate into things like nerve cells. Whilst it holds a lot of potential, that's then stopping a potential embryo from surviving. So there's a lot of ethical concerns around that. Most recently, CRISPR is a modern DNA technology, and it's to do with genetic engineering, modifying of the genome. And this is really sort of very new age, because we're actually now looking at how can we cut pieces of DNA out or insert genes. This could have a lot of potential in looking at how we can reduce diseases that are caused by genetic factors, mistakes in the DNA. So there's so much potential with it, but where do we draw the line? Are people going to start selecting for genes that are just more desirable? Are they going to remove genes because they just don't like that? there's a lot of issues around that. So in your tasks that I'll get you to do, you're going to have a little bit of a look at this a bit more. This is an interesting quote when we start thinking about genetics, and this quote that I found by Greeley states that the new genetics refers to the increase in power of genetics that followed understanding of the biochemical basis for genetics in DNA. So we had this understanding, but there's now an increase in power that's come from that. This heightened power both creates new ethical and social concerns and exacerbates older ones. So while our understanding is continuing to increase, there's now a whole lot of new concerns that come with that. There's a video that I'd like you to watch on the PowerPoint that you can click on in your own time. Now, thinking of your RI, how can we relate this to the claims at the beginning? A quick Google search of some of these things, and I found a number of journal articles. So this one here looks at the bioinformatics toolbox for reproductive biology. So how can bioinformatics be used in this sense? This one here is looking at the evolving role of genetic testing in reproductive medicine. So looking at that pre-implantation screening sort of stuff. Okay, another one from a more genetics perspective, looking at assisted reproductive technology and functional genomics. So this was a study looking at farm animals and embryos that have been produced through IVF and cloning. So you can have a look at some of that sort of stuff. This one here was looking at assisted reproductive technology with genetic modification, which is that last claim. So lots of things here that you can relate back to reproductive technologies. So this lesson, you needed to be able to discuss some of the implications of genetic screening technologies. So thinking about pre-implantation genetic diagnosis and CRISPR on the reproductive technologies. Now we're going to investigate this a little bit further in the tasks that you're going to work through, and I'm going to be having a look at what you've come up with from this. So see the class notebook for what I'd like you to do. You're going to be watching a click view looking at CRISPR and reading an article about pre-implantation genetic diagnosis.